Hello and welcome to another episode of Research Talk. Today we have with us Dr. Shubham Sahu. He's from Kanpur, Uttar Pradesh. He completed his BSc at Banaras Hindu University and completed his integrated PhD from the Indian Institute of Science, Bangalore. He's currently a postdoctoral research associate at JNCSR, Bangalore, and his research focuses on the biomedical applications of synthetic entities. In today's episode, he will be talking about the organic nitrite donors as nitric oxide prodrugs. So, Dr. Shubham, welcome to the talk, and the screen is yours.
Okay, so first of all, thank you, Mahesh, for the kind introduction and giving me this opportunity to present some of my work, which I have done during my PhD. So in this talk, I will talk about nitric oxide and nitrite and how these molecules act as biologically active molecules and how they are helpful in various biological processes, especially cardiovascular health, heart health. And in this talk, I will talk about the synthesis of some of the prodrugs, which are like organic molecules, which release nitrite in cellular conditions, and that nitrite gets converted into nitric oxide and exerts its biological significance. So let me start with nitric oxide. Nitric oxide plays several important roles in human body, and here I am presenting mainly three of its major roles. The first one is that it acts as a messenger molecules. In neuronal cells, it sends information from one neuron, one synapse to the other one. That's why it is called a neurotransmitter. And second one is the killer in macrophage. Basically, this is like for killing cancer cells or any foreign object in the body. So this kind of NO is released in the body only when it is required. And that's why it is called INOS, inducible nitric oxide synthase enzyme, which produces this nitric oxide. And the third one, which will be the main focus of today's talk, is the endothelial nitric oxide synthase and the nitric oxide produced by this enzyme. So basically it is produced by the blood vessels. As you can see here, let me show the using the pointer. Yes, so this I am showing a schematic cross section of a blood vessel. So this enzyme enos is present in the innermost layer, which is called endothelial cells, where it is continuously produced. It produces nitric oxide, and blood pressure is regulated by regulating the diameter of the blood. So suppose you are running, so more and more blood flow is required. So in that case, the diameter of the blood vessel has to be increased to sustain a optimum blood pressure. So that's the one sentence explanation for this. Now, this is the healthy condition. Now, as we age, there will be several problems, and as you can see here, the human cardiovascular health is deteriorated with age, and most probably this is the deposition of fatty acids, sorry, fatty deposits or any salts like calcium and all, which block the flow of blood in the blood vessels. So now this can be due to various factors like diabetes, high blood pressure, high cholesterol in the blood vessels, or physical inactivity or unhealthy lifestyles such as tobacco consumption or alcohol consumption, or due to various other factors such as age, gender, or genetics. So what two of the major problems are here that if this blockage happens in the blood vessel which carries blood to the brain, it is called stroke. Now, if it happens to the blood vessel which is connected to heart and other part of the body, it is called heart attack. Now, under these conditions, NO-based therapeutic is required. So now that's why over a period of time, people have tried to develop different types of NO donor molecules. And this was in 1980s that it was found that NO acts as a signaling molecule, and over the period of time, it has been shown that various molecules like simply this salt can release NO, nitrosothiol derivatives, or hybrid compounds like which have dual function, like this moiety releases nitric oxide and this acts as a anti-inflammatory molecule like N-acetyl salicylic acid is there. This is salicylic acid derivative basically. So methyl ester. And there are several other like polymeric materials and photosensitive NO donors, pH sensitive NO donors have been reported. Now, because NO is a gaseous molecule, so it exerts a lot of side effects. So that's why presently the focus is shifted on the development of nitrite based therapeutic and synthetic nitrite donors. Now I was talking about nitric oxide and how it is generated. So chemically, if we see, this is generated in human body by using L-arginine. It is an amino acid, essential amino acid. So basically, it's the two-step oxidation by the enzyme called nitric oxide synthase, and this produces nitric oxide. So why nitric oxide is so reactive, and it is very important to regulate its delivery to particular location in body wherever we want its positive impact. Now, if we look at the molecular diagram of nitric oxide, it has one electron in the anti-bonding pi orbital. Now it has very high energy, so it can easily either give an electron and get stabilized, so it will convert into NO plus, or it can accept another electron, or it can get into convert into NO minus. Now that's the origin of its high reactivity, and also so therefore, it has the ability to react with various type of biomolecules present in human body, and it can also generate highly reactive peroxynitrite species that can damage the intracellular proteins, DNA, and many other things. So that's why localized delivery of this gas is very important. Now here comes the selectivity thing. Therefore, it has been recently discovered, like last 15 years or so, it has been found that nitrite acts as a alternative in human body. Now, when nitrite acts as alternative to NO, when at low concentrations, at normal oxygen concentration, NO is produced by the enzyme which is present, as I discussed, in the endothelial cells, and it delivers its effects via enzymatic pathways where it leads to vasodilation. Basically, vasodilation is the regulation of blood pressure by regulating diameter of the blood vessel. Now, when the oxygen concentration goes down, NO is not produced by the normal pathway by the enzyme. Now, in that case, whatever nitrite is present in human blood, that is used as a source of nitric oxide. So, nitrite can do the same work what NO does in 2 to 3 different pathways. One is like it can inhibit an enzyme known as catalase, which is produces, once it is inhibited catalase, it produces S2O2. So which through various pathways leads to the same effect as nitric oxide. And another thing is that there are proteins like which can convert nitrite into nitric oxide, and they can exert the same effect. So basically, human body, actually it is known to balance everything, and as you can see here, there are proteins which can convert nitrite to NO, and also if there is excess NO, it can be converted into a nitrite using another protein called ceruloplasmin. So that's the basically origin of developing nitrite based therapeutics. First is that it is naturally available in human body. Second is that there are mechanisms which can convert nitrite into NO. So that was the start of this our project, and we had a hypothesis. We were looking for compounds which can release nitrite. So we came across a set of molecules called 4-nitronaphthalene-based molecules. We found that these molecules have been used to sense thiols. Now we wondered what happened to this nitro group when it senses thiols. So we speculated that something like this might be happening, that thiol reacts with nitrite in cells, and this nitrite is released, and can it release nitric oxide using the proteins which are available in body. So this was the starting point for us. Now there are very few reports about nitrite donors, and this field is like at the, it is emerging, and it is growing stage. So if you see, there are like two different type, like allylic nitro derivatives and this benzyl based nitro compounds are there, which are able to release nitrite. Now first two have no relevance to human body because this can be released by the thiols, whereas this third one requires dithiols. So basically you can see that research is just started in this area, and we need to develop like a different kind of different type of molecules which are able to release nitrite and compare their therapeutic capability. So once we had hypothesis, we wanted to test it whether these kind of molecules can indeed release nitric oxide in cells. So we synthesized first only one molecule and tested that these molecules were indeed able to release nitric oxide. So whatever you are seeing the green fluorescence, that indicates that this is corresponding to nitric oxide. So this we test using a well-known probe called DAF-FMDA. So this is a fluorescent probe. When it's stored inside cell, if NO is there, it will react with NO in the presence of oxygen and produce a fluorescent product DFFM triazole. So that's what we are seeing here. So this confirmed our hypothesis that indeed these 4 nitronephthalimide derivatives are able to release NO, and we wanted to see what is the mechanism next. So we synthesized like several type of different structure relationship having that idea in mind, and then we selected few of these derivatives to test to the next experiments. So the synthesis were very straightforward, that we started with the AC naphthene, where we nitrated it nitrated it, and we got like 5 nitro AC naphthene, and which was oxidized to get this anhydride. In the second step, we wanted to synthesize at the 3 position to establish a structure activity relationship. Now here we found that one important thing to notice in this synthesis is that here first we do oxidation, so that electron withdrawing group now will direct the nitration to the meta position, whereas first where we do nitration so that it can go to 4th position. So this was the synthesis part. So we synthesize several derivatives, and we synthesize one compound without nitro group to just see the effect is just because of the nitro group, not other part of the molecule, as a negative control 2.11. And we have characterized fully these molecules like proton 13C, NMR, HRMS, and we got single crystal for 4 nitro derivative and 3 nitro derivative, one compound from each series. Now this was the synthesis and characterization. Now we went ahead to check first step whether these compounds are able to release nitrite. Now as I explained that this kind of compounds have been known for sensing thiols. So we started with the most biologically abundant thiol, GSH, and we found that indeed these compounds were able to release nitrite using this GSH as a nucleophile, and also whatever the product is formed, this is called GSH conjugate. So with that is fluorescent, because now this sulfur is electron donor. Now this donor acceptor charge transfer process happens, and this gives fluorescence, whereas you can see 2.1 starting metal is not fluorescent. So basically we found that all the 5 nitro derivatives were able to release almost 100% nitrite quantitatively, whereas 3 nitro derivatives were not, as you can see here. And the formation of this GSH conjugate and nitrite was 1:1 here. You can see the green fluorescence corresponds to this thiol conjugate, whereas the pink one is amount of nitrite. So we had varied like amount of compound and thiol. We see the 1:1 correlation, and we also checked the different thiols, some of the biological thiols and some with different nucleophilicity. So we also found out that this 4-nitrothiophene, where this SH thiol is least nucleophilic, because once it goes S minus, because of a strong electron withdrawing group, it is highly delocalized and it is not available for nucleophilic substitution reaction. Now the next step, the selectivity is very important in the biological context. So we wanted to see whether this reaction is only catalyzed by the GSH or any other amino acids present in human body or salts. So we found that it was highly selective to GSH as compared to other amino acids like glycine, proline, methionine, serine, and arginine. Similarly, these are the components which are present in human blood, which could interfere, but we found that afterwards reaction was highly selective. And further, what we did, we methylated the free thiol of the octane thiol and saw the reaction. So we saw that negligible nitrite release was happening. So from here we concluded that free thiol is very important for nitrite release, and these compounds are highly selective towards GSH, especially thiols. Now as I explained that 3-nitro was not able to release nitrite. So this is very simple, is in the sense that this is an aromatic nucleophilic substitution reaction. So here what happens, the nucleophile attacks at the nitrocarbon, and then this leads to the formation of Benzenheimer complex. Now this again, this complex here, the carbon nitrite bond breaks, and it leads to formation of thiol conjugate and nitrite release. So to explain this, if we draw the resonance hybrids of 4-nitroethylene, we will see that positive charge comes at the C4, C2 positions. So, and when compare with C3, positive charge doesn't come at this meta-nitro carbon, that indicates that these two locations are more prone for nucleophilic substitution as compared to the C3. So that explains the reactivity. Also, we had done some theoretical calculation to check the charge. We found that 4-nitro had 10 times, almost 10 times much higher charge as compared to 3 groups. That also supports that 4-nitronephthalamides are more reactive towards nucleophilic substitution. And also the same fact was checked by doing a reaction. So we did a reaction of 4-nitronephthalamide with octane thiol. Under DMF condition, it went in room temperature with a gold yield of 70%, whereas this 3-nitro reacted only at very high temperature and gave an yield of very less 5%. That also tells that the 3-nitro is very much inert towards the nucleophilic substitution. So I have talked about the measurement of nitrite release from this compound. So for measuring the amount of nitrite released, we have used a well-known assay called grease assay. So it is a combination of two components. One is 4-amino sulfanilamide and N-naphthyl ethylene diamine. So what happens in the first step, nitrite under slightly acidic conditions, this slightly acidic conditions are maintained in this reagent so that diazotization of this amine happens, and this reacts with this N-naphthyl ethylene diamine to give this pink azo dye. Now, as you can see here, when we add sodium nitrite to this reagent, it gives absorbance corresponding to this pink azo dye at 540 nanometer. Similar absorbance is observed for the compound reacted with GSH. Now, for quantification, we have separately obtained a calibration curve. So where absorbance of this dye formed is plotted against this sodium nitrite known concentration. So if we have our test condition, any absorbance we can correspond to the particular concentration of released nitrite. Now in the second step, as I was talking about nitrite to NO conversion, organic nitrites as NO prodrugs, it was important to understand how these molecules are releasing NO from nitrite. So myoglobin, actually it is a iron based protein which is well known to reduce nitrite into NO. So and also it is commercially available. So we wanted to use this and study the mechanism of NO release from nitrite. So commercially available myoglobin protein is in iron 3 form. It needs to be reduced to iron 2 form so that it can react with nitrite. Now these three species which are formed after the reaction, after the subsequent steps, they have distinct UV absorbance, like 409 nanometer corresponds to this metamyoglobin, 434 corresponds to this iron 2 form, and after it has reacted with the nitrite and NO is released, and this released NO is bound to myoglobin, this gives this distinct peak as 421 nanometer. Now corresponding this Q peaks are also observed, which indicate the distinction between the different species, and myoglobin nitrosyl corresponds to this 550, 580, this humps. And we have compared it with just reacting with sodium nitrite, and our compound 2.1 reacted with GSH. So that confirmed that nitrite is getting converted into NO using iron-based protein. Now the next step, we wanted to see the HPLC based reactivity. So here we reacted all the 4 nitro derivatives with GSH and saw that the single peak complete conversion was happening, and this GSH adduct was forming, which was confirmed by HPLC and ESIMS. Now again, this 3-nitro, as I discussed, they were least reactive and we could not see any product under the 12-hour condition. We also wanted to see how these compounds react with a mixture of thiols. So we combined all biologically relevant thiols like GSH16 and acetyl-cysteine, mixed them and reacted with this compound 2.5, because in this case peak separation was well resolvable. So we could see that GSH adduct is the most amount it is forming in quantity, and this we could see by reaction with separate. As you can see, 16 gives 2 peaks, so that its complete conversion is not happening, whereas cysteine has higher reactivity than compared to cysteine but less than GSH. Now we have checked this NO release in cells, so we found that these compounds release nitrite. This nitrite gets converted to NO using iron proteins, and we were able to see. Now we have also isolated the what is forming inside the cell. So whatever this green fluorescence you are seeing, that corresponds to NO, and this blue one basically, actually it is green, but to distinguish this pseudo color is used. Now this is isolated cells. Cells are like isolated, and they are lysed basically to see what is their component, and after evaporating the aqueous phase, we will dissolve the component of the cells and analyze, and we found that all these 5 compounds which have like nitro group at 4 position are able to form GSH conjugate in cells, and whereas this 3 nitro is not able to do so, and this we confirmed by HRMS spectroscopy, as well as separately synthesizing all these conjugates through RB flask reaction, and we got the peak at the same position which we got in the from the cells. And when we did like concentration dependent studies in HeLa cells, we saw that there was an increase in like 1 micromolar to 5 and 5 micromolar. There is, you can see there is an increase in the fluorescence of both. Like here I'm showing for 2 compounds. The blue is glutathione conjugate, green is the NO detection. So you can see, and this compound 2.2 was found to be the best in NO releasing. You can see it has gave the highest intensity of its GSH conjugate as well as NO release. Now to check whether this NO is indeed released, there we used alternative method. There is this called PTIO, this compound. It can scavenge this radical nitric oxide. So basically what happens, as you can see here, when the cells were treated with these 2 compounds, they gave the good amount of NO release as indicated by the fluorescence of the dye probe. Now when we incubated along with this scavenger, this is drastically decreased. That clearly indicates that these compounds are indeed releasing NO, and this reactivity was actually selective to GSH only, and across different pH, this methionine and octane thiol with the methyl protected could not release nitrite. So that also indicates that the reactions are again very selective to GSH. Now, so there are another kind of cells, as I was talking about the endothelial cells. So basically these endothelial cells are isolated from the umbilical cord of the newborn baby. So if you look at the microscope, they look like this, and this is a schematic. Now this HUVEX, human umbilical endothelial cells, they are actually called primary cells because they are directly obtained from a living system and used for study, and they are like widely used in medical research because they are act as a model for blood vessel lining. I was talking about in the introduction section that endothelial cells where the enzyme is present, they produce nitric oxide. So to test our compounds in actual more relevant cell conditions and more relevant to human body, we used these cells, and these cells were taken from hospitals with ethical clearance from the Institute Human Ethics Committee. Now, in these cell lines, what we did, we incubated our compound and saw that there is a time-dependent increase in the fluorescence of both. The blue fluorescence is GSH conjugate, and the green fluorescence corresponds to the NO release. Now, after 40 minutes, you can see this blue channel is the fluorescence of the GS conjugate and this NO release. So we were able to test our compounds in the more relevant cell lines, and indeed these compounds are able to release nitric oxide. So now if I want to summarize this, so we found that this 4-nitronephthalimide class of compounds are able to release NO in cells through nitrite release, and NO production was happening by iron proteins. And also the structure-activity relationship indicated that these compounds, only 4-nitronephthalamides are selective to nitrite release, not the 3-nitro ones. And there was another interesting study we did in this case, that because the product of the reaction of this compound with thiols in cells was fluorescent, we were able to distinguish how much amount of NO is coming exactly from the our compounds. And because these cells which we used like endothelial cells I just talked about, they also produce nitric oxide. So basically we have two kind of nitric oxide when we incubate the cells with compounds. One is coming from the natural enzyme present in the endothelial cells, other one is from our compound. Now when we use this commercial probe, it gives us a combined intensity, and in this you can't distinguish how much NO is coming from compound, how much is coming from the natural enzyme. Now because we are having our side product which is forming after release of nitrite, that is also fluorescent. Now we correlated the fluorescence of this dye intensity and this GS conjugate, and with a mathematical equation we were able to measure the relative amount of NO levels which are coming from the cells as well as from the compounds. So this is the completion of my talk. So I would like to thank you for your attention and this opportunity again.
Nice presentation, Dr. Shubham. Nice work and nice presentation.
Thank you, Mass.
If you enjoyed this session, don't forget to like, share, and subscribe. We'll meet you in the next episode, next session. Thanks for watching.